Now we can look at genes, proteins, and RNAs, which have a unified search menu. Let's click on that. We have a separate area for gene names and protein names and EC numbers. Let's start up in gene names, and I'll start typing THR again, and you'll see it actually offers me an autocomplete to a number of gene names. Handy. Now, let's move down to protein names. If I start looking for my autocomplete there we go. It gave me a bunch of options, and I could keep typing and it would pick the best one. Let me clear my search up here just to keep from confusing it. You also, once again, have the option of a number of searches or filters down here. It's a big range. We can search or filter by sequence length, replicon, product molecular weight, isoelectric point, regulators, evidence codes, cell components, gene ontology terms, multi-fund terms, and even publications. Now, you're really going to want to play with this on your own, but let's take a look at screening by combining two terms. For example, we could start by looking at molecular weight, and we want something that's between 60 and 80 kilodaltons. Next, we can go down to the cell component. Now, this gives us access to the cell component ontology, which is an ontology that's an ordered hierarchy that describes parts of the cell. And let's pick a very high-level term, membrane. So now, when we submit the query, we're going to be asking for a list of all the E. coli membrane proteins between 60 and 80 kilodaltons. Let's do that. Okay, so it turns out there are 53 E. coli membrane proteins of some kind, that's inner membrane, outer membrane, that are between 60 and 80 kilodaltons. Again, our results come to us on a sortable table where we get the gene name, its product name, its location, since we asked about location, and its molecular weight. Okay, let's do one last look at this page. Once again, we can search by sequence length, replicon, so say if something has two plasmids, is this on one of the plasmids? Product molecular weight, isoelectric point, regulators, cofactors, substrates, and so on, evidence code, cell component, gene ontology term, multi-fund term, and publication. Now, let's take a look at searching by reactions. Once again, and as always, we can search by name. In this case, we can also search by enzyme commission number if you happen to know what enzyme commission number you're looking for. We can also search or filter by ontology, and this is actually going to dip into the enzyme commission numbers in a structured way that you can browse through, or by reactants or products. And again, autocomplete will come to our aid. We start typing and oh, yes, we wanted L-threonine. Now it's important that you actually pick a specific name here because the compound search accessing the reactions here needs the exact name of the compound. It's not a substring search against the compound, it's actually looking for that exact compound in the reaction because that avoids problems with getting a giant list of reactions when you have the substring for the compound. So if you're not sure exactly what the compound is, let the autocomplete help you out, since it has access to our full list of compounds. And again, if I submit the query here, we get another table. This shows us the reactions, and when present, the EC numbers. And again, this is a sortable table. You could, for example, sort by EC number. So that's what the reaction search does. It lets you search for reactions by name, or it lets you search for the reactions by compounds that are involved in those reactions as either products or reactants. The last intermediate search we have is the pathway search. Again, the pathway search is similar to our other structured searches. You can search by name. You can also search or filter by our pathway ontology, by the number of reactions, by substrates present in the whole pathway, by evidence code, and by publication. So for example, let's try combining two of these. Say we're once again interested in a pathway that involves L-threonine. Seems good, right? And say we want to filter for just pathways that were inferred by direct assay. Now, I'm not going to go into detail about what the evidence code means here, but we have descriptions on the BioCyc main page, we'll link from there, about what our evidence codes mean. The short version is that an evidence code explains what kind of basis we had for asserting that something is present or operates in a certain manner. In this case, the evidence code is the literature-derived experimental basis for saying that some pathway that we're about to find that uses L-threonine is present in E. coli. So let's see what we can find. Okay, so it looks like we have 8 pathways that involve L-threonine that are present in E. coli that were inferred from a direct assay. Interesting, right? The table reports out to us the pathway names, the number of reactions, and their evidence, which of course is all the same because we asked for a certain kind of evidence. And we can click through on any one of these things to the pathway. We can click through to an explanation of what this means for the evidence. And down below here we actually have your query converted into our BioVelo query language, which is the advanced query language. So if you're going to get involved in learning the advanced query language and you really want to do it in free query language form, this will be helpful to you. But otherwise, you can just ignore this. It's for educational purposes.